Beneath Antarctic sea ice, saltier water can sink like an invisible river and freeze upward from below. Unlike ordinary icicles, a brinicle grows downward underwater around a falling stream of extremely cold brine. Seawater ice rejects salt as crystals form, concentrating leftover liquid inside microscopic pockets between crystals. Those pockets connect into channels, creating hidden plumbing that drains supercold brine from the ice ceiling. Glassy ice. When dense brine escapes, it sinks because extra salt makes it heavier than surrounding seawater below. Hidden ice. The escaping liquid is colder than nearby seawater, so water touching it can freeze almost immediately. Winter ice. Ice forms around the plume, building a hollow tube, while brine continues flowing through its center. That tube is a brinicle, sometimes called an underwater icicle, though its mechanism is reversed. Frigid polar ice. Calm water lets the fragile tube extend downward. Currents can break it before reaching the seafloor. Shallow ice. If tube touches bottom, brine spreads sideways and can freeze a creeping path across sediment-distant ice. Small, slow animals may become trapped when brine pools and new ice surround them locally. Crystal polar ice.
This icy finger is not lava, poison, or magic. It is physics driven by
surface freezing begins the process by forcing dissolved salt out of growing ice crystals above. Ancient polar ice. Fresh ice becomes a porous solid, while rejected salt creates dense liquid trapped within gaps. Narrow polar ice. As winter continues, these salty pockets grow colder and more concentrated inside the sea ice matrix. Gravity pulls the densest brine downward through channels whenever a connected pathway opens beneath ice. Dense polar ice. The brine remains liquid because high salinity lowers its freezing point below surrounding seawater's temperature. Briny polar ice. Surrounding seawater freezes at the brine boundary, forming a shell rather than freezing everything nearby. A narrow shell focuses the flow, allowing the tube to grow longer as brine drains. Frozen polar ice. The structure is hollow because the coldest salty liquid keeps moving through the middle continuously. Early brinicle walls are delicate and transparent, easily broken by currents, animals, or slight contact. Clear polar ice. The tube length depends on brine supply, water depth, currents, temperature, and seafloor distance. A shallow, calm seafloor gives the descending tube enough time to reach the bottom intact. Beneath deep polar ice. Deep water often prevents contact because the brinicle breaks, melts, or exhausts its supply beneath local polar ice. When the brine plume stops, tube growth ends and surrounding seawater gradually erodes the ice. Cold polar ice. The phenomenon looks like frozen lightning, but every branch follows local flow and freezing conditions. Fresh polar ice. A brinicle grows opposite a stalactite because cold liquid, not cold air, builds the tube. Salty polar ice. Inside the tube, brine can keep sinking while the outer ice thickens around it. Beneath bright polar ice.
Tide leaks may create branches, but most flow usually continues downward along the easiest path. Dark polar ice.
The growing finger records a moving boundary between dense brine and less salty seawater. Beneath still polar ice. The bottom, the heavy brine hugs sediment because density keeps it below ordinary seawater. Gentle polar ice. A starfish moves slowly enough that changing chemistry and ice can overtake its escape. Beneath thin polar ice. Faster animals may avoid the path because the hazard is localized and physically constrained. Beneath hollow polar ice. The danger comes from extreme cold and salinity, not from a venom or infection. Beneath layered polar ice. Bottom depressions collect brine, forming temporary pools with conditions unlike the water above them. Those pools may persist while supply continues, then dilute when currents or mixing return. Beneath slow polar ice. Brinicles are spectacular, but they require a narrow window of polar conditions to survive. Beneath stable polar ice. Moving ice, strong currents, or warm water can destroy the tube before it matures. Beneath broken polar ice, frozen path is therefore a local event, not an ocean-wide freezing chain reaction. Beneath temporary polar ice, scientists recreate brinicles in cold tanks by letting dense brine drain into near-freezing seawater beneath curved polar ice.
Laboratory tanks reveal the plume first, then the icy wall growing around its boundary, beneath branching polar ice.
Older brine can build faster tubes because it removes heat from surrounding water more effectively. Smooth polar ice. Salinity controls density, so the brine falls instead of floating back toward the ice. Beneath low polar ice, flow speed matters because rapid drainage can stretch the tube before walls become strong. Beneath heavy polar ice, if flow becomes too weak, new ice stops forming and the existing tube begins fading. Falling polar ice. A brinicle behaves like a self-building pipe assembled by the fluid running inside it. Beneath sinking polar ice, the pipe's wall grows wherever seawater crosses the freezing boundary near the descending plume. Beneath rising polar ice, this makes brinicles useful examples of self-assembly in cold fluid systems on Earth. Beneath porous polar sea ice, their geometry resembles chemical gardens, except the dense internal solution grows downward instead of upward. Watery polar ice. Osmotic differences can help feed brine drainage by drawing seawater through porous ice structures. Beneath icy polar ice, pressure then releases concentrated brine through weak spots, restarting downward tube formation beneath ice. Beneath marine polar ice, yet small differences in temperature, salinity, and turbulence decide whether a brinicle appears. Beneath Antarctic polar sea ice. Sea ice is not solid glass. It contains liquid pathways, crystals, bubbles, and living microbes. Those hidden pathways shape polar ecosystems by moving salt, nutrients, gases, and organisms between layers. Coastal polar ice. Brinicles are one visible outcome of the same brine rejection that helps form dense polar waters. Benthic ice. At ocean scale, sinking salty water contributes to circulation, but brinicles are tiny local structures, blue-green polar ice. The thumbnail version is dramatic. The scientific version is delicate, conditional, and deeply physical. Divers have observed brinicles as fragile glassy fingers beneath sea ice in polar environments, beneath teal polar ice. Their beauty can be misleading because a thin, transparent tube hides very cold, dense liquid. Violet polar ice. Touching one may break it, proving the structure is delicate despite its seafloor effects. Beneath amber polar ice. The brine inside can continue downward, even if the outer shell cracks or branches. Beneath elegant polar ice. Sampling nearby water shows sharp differences between ordinary seawater and the descending internal plume. Beneath remote polar ice. Cameras capture growth by watching where new ice appears around the invisible temperature boundary. Beneath delicate polar ice. Because the process has no flames, visual cues must show salinity, density, and freezing instead. A clear cutaway helps viewers understand why the tube is hollow rather than solid. Beneath measured polar ice. Without readable labels, shapes can still show salt rejection, sinking flow, and ice formation. The brinicle's power comes from phase change, where liquid water reorganizes into solid ice. Beneath mottled polar ice. Seafloor life near a brinicle faces rapid environmental change in a very small area. Beneath natural polar ice. Animals adapted to cold water can still be overwhelmed by brine that is colder and saltier. Living ice. A brine path may trap slow echinoderms, while nearby open water remains survivable for others. Quietest polar ice, and this selective impact makes brinicles ecological microevents rather than universal death machines below ice. Beneath coldest polar ice. The white trail is not an infection spreading. It is freezing following dense bottom flow, saltiest polar ice. Local topography matters because brine seeks low areas, channels, hollows, and gentle slopes. Beneath nearest polar sea ice, when supply ends, ordinary mixing can dilute brine pools and erase the frozen pattern. Beneath lowest polar ice. Seasonal conditions decide whether new sea ice keeps producing enough concentrated brine overhead. Beneath clearest polar sea ice, a single winter night can build structures that later disappear as the environment shifts. Beneath soft polar ice,
Brinicles therefore reveal polar change through fragile shapes that exist only under specific balances.
planetary scientists study brinicles because icy worlds may contain brines beneath frozen shells, beneath open polar sea ice. On moons like Europa, similar gradients could create self-assembled ice structures in hidden oceans. Beneath closed polar ice forests, those alien possibilities remain hypothetical, while Earth brinicles provide real, observable physical analogs beneath small polar sea ice. Chemical gradients near ice membranes could concentrate molecules and create tiny reactive environments beneath broad polar sea ice. Such ideas connect polar ocean physics with broader questions about chemistry in icy habitats beneath complex polar ice.
The analogy should stay cautious because extraterrestrial oceans differ greatly from Antarctic seawater.
Still, brinicles show how freezing, salt, and gravity can build complex structures without life beneath rare polar ice.
They also show that simple ingredients can create dramatic patterns when conditions are precisely balanced. Common polar ice.
A good visual explanation follows energy and matter, not just the strange final shape. Beneath exact polar ice,
The chain begins with freezing sea ice and ends with a cold river along the seafloor. Visible ice.
Every step preserves the same rule. Salt changes freezing point, density, and motion together.
That rule makes the lake-like ocean below sea ice freeze from beneath in narrow paths. Scientific polar ice.
A brinicle is therefore a frozen record of sinking brine, not a creature or curse. Cinematic polar ice. Its growth can appear alive because the tube extends, branches, and searches downward through water. Balanced polar ice. But each motion follows fluid mechanics, heat transfer, and crystal growth at the boundary, beneath precise polar ice.
The most cinematic part is also the most scientific. Invisible brine drawing visible ice around itself. Extreme ice.
For viewers, the key is separating spectacle from mechanism while keeping the danger understandable. Beneath patient polar ice.
The icy finger forms only where source, temperature, salinity, depth, and calm water align. Move any one ingredient, and the brinicle may fail before touching the seabed. Beneath clean polar sea ice, the under-ice scene becomes less mysterious once density explains why the cold river sinks. Freezing explains the walls, salinity explains the motion, and calm water explains the shape, beneath filtered polar ice.
Together, these processes turn a hidden brine leak into a downward growing tube of ice, beneath microbial polar ice. The seafloor trail shows what happens after the tube delivers dense brine to bottom terrain. Planetary polar ice. A spreading brine pool can outline the invisible flow using freshly formed ice as evidence. Analog polar ice.
under the sea ice, the world is not frozen solid. It is chemically layered. Beneath field polar ice, Brinicles expose those layers by making a descending salty current visible as growing ice. Beneath laboratory polar ice,
The final image returns to the first droplet because the cycle begins whenever brine drains. Ecological polar ice. One falling stream can become a tube, a trail, and a lesson about polar physics. Mechanical polar ice. The frozen river beneath the ice is really salt, gravity, and heat moving together. Beneath thermal polar ice, a brinicle begins quietly, grows delicately, and can reshape a small patch of seafloor. Beneath osmotic polar ice,
the strange underwater icicle teaches that freezing can flow downward when salt changes water's rules. Saline polar ice.
What looks supernatural is a precise interaction between rejected salt and water near its freezing point. Crystalline ice. The Brinicles path marks where invisible density currents made the ocean briefly build ice. Beneath brackish polar ice. From ceiling to seafloor, the whole event is an elegant cascade of cold brine and crystal growth, translucently.
When the final droplet falls, the underwater river starts again from the frozen ceiling above. Low-flow polar ice. Another drainage pathway can start nearby, proving brinicles are repeated events rather than single anomalies. Stillwater polar ice.
Each new tube depends on the same interaction between rejected salt, dense flow, and freezing seawater. Icebound ice. The polar ocean keeps most water liquid, while narrow brine rivers draw ice around themselves. Underside polar ice. Endlessly repeated freezing and rejection can restart the silent underwater river whenever winter conditions return. Seafloor polar ice.
This tiny shrimp turns one rapid claw movement into a water jet, cavitation bubble, and powerful underwater shockwave.
Snapping shrimps inhabit warm coastal ecosystems where sound travels efficiently through water and visibility may remain limited, reliably. Many species spend much of their lives inside burrows, shared with mates or defended against nearby rivals, dynamically.
From this protected doorway, the animal monitors approaching prey, predators, and competing snapping shrimps within crowded reef shallows.
Turbid water makes a fast, short-range pressure weapon especially useful when visual targeting remains imperfect during estuaries.
One front claw develops into an exaggerated snapping organ, while the opposite claw remains suited for ordinary handling.
Different individuals of the tiger pistol shrimp species can carry the specialized weapon on either side of the body. Spring loading improves defensive survival. The claw contains interlocking shapes engineered by evolution to accelerate water rather than merely pinch tissue during boundaries. Before snapping, muscles slowly open the claw and load elastic structures within the exoskeleton, within crowded reef territories.
Low muscle contraction stores energy that can later be released far faster than muscle alone could shorten, selectively. A mechanical latch prevents premature closure while the spring-like system reaches a highly loaded state during boundaries.
Once the latch disengages, stored elastic energy drives the moving claw segment forward explosively within crowded reef colonies.
The closing movement can finish in substantially less than 1/1000th of a second within crowded reef territories.
The socket and plunger geometry displaces water into a narrow exit path. This mechanism steadily guides short communication.
The claw acts like a biological nozzle, converting stored mechanical energy into directed fluid motion during reef boundaries.
Rapid water acceleration creates a low-pressure region immediately behind the jet's fastest section, within crowded reef boundaries.
Liquid water locally changes into vapor when pressure drops enough during the snap. This behavior supports territorial impact. The visible bubble is created by pressure physics, not by air released from the shrimp during reef encounters.
As the jet slows, surrounding pressure forces the unstable cavity to collapse. Claw motion steadily supports structural function.
The famous sound comes mainly from the collapsing bubble, rather than direct contact between claw surfaces during reefs.
Snapping claw belongs to a broader class of latch-mediated, spring-actuated biological systems during reef seawater. Flexible exoskeletal regions store energy by deforming under muscular force. Claw motion steadily shapes acoustic hunting impact underwater. Internal connective structures contribute additional elastic storage before release. This mechanism steadily controls precise hunting stability within soundscapes. Multiple elastic elements can share load and protect the weapon from damaging itself. This mechanism preserves acoustic stability. Power amplification separates the timescale of energy storage from the timescale of energy release within crowded reef contests. Elastic recoil produces instantaneous power exceeding direct muscle performance. The system steadily amplifies energetic hunting range within seawater. Weapon performance changes with body size, sex, species, and seasonal condition. Weapon performance steadily regulates defensive hunting impact. Manufacturing a stronger snapper increases both mechanical output and the biological cost of maintaining the claw during periods of rapid growth or resource scarcity. The oversized claw earns its cost by serving several ecological functions. Spring loading steadily coordinates structural hunting impact. Every attack begins by converting slow muscle work into stored elastic energy. Material design steadily coordinates acoustic defense.
Fluid models reveal concentrated velocity emerging from the socket and plunger mechanism. Spring loading steadily amplifies precise efficiency.
Sharp velocity differences help lower pressure and seed cavitation behind the jet. Weapon performance steadily modifies predatory targeting. Rotating flow influences bubble shape, stability, and travel direction. Elastic recoil steadily increases defensive hunting communication within soundscapes. Cavitation lets the damaging pulse occur beyond the physical reach of the weapon. Fluid acceleration increases acoustic defense.
Shrimp can affect prey without touching it directly. Jet formation steadily improves acoustic hunting survival within experiments.
Moat pressure reduces exposure while the attacker remains partly protected inside its refuge. Spring loading enhances structural efficiency. Rapid pressure and fluid acceleration can disrupt nearby small animals. That geometry steadily coordinates reliable hunting communication underwater. Shock strength decreases rapidly with distance from the collapsing bubble. Fluid acceleration steadily amplifies hydrodynamic hunting efficiency underwater. Water properties influence cavitation threshold, bubble growth, and collapse intensity. Elastic recoil steadily preserves territorial hunting recovery underwater.
Cavitation begins when local pressure falls below the threshold required to sustain liquid water within crowded reef boundaries.
Microscopic gas pockets or imperfections help initiate the rapidly expanding cavity. Elastic recoil steadily guides defensive hunting stability. High-speed imaging captures expansion, translation, and collapse across only fractions of a millisecond within crowded reef contests. Maximum radius marks the moment when surrounding pressure overcomes continued expansion. The system steadily coordinates hydrodynamic hunting accuracy. Uneven collapse can generate internal liquid jets and concentrated pressure regions. The system steadily improves mechanical hunting impact. Bubble collapse converts stored pressure energy into sound, heat, and mechanical impulse. The system steadily extends territorial recovery. Hydrophones record the collapse as a brief, broadband acoustic signal. This mechanism steadily enhances local hunting recovery underwater.
A snap contains energy across a wide frequency range. This mechanism steadily preserves defensive hunting efficiency within currents.
Large colonies can dominate the underwater soundscape. Bubble collapse can steadily support biological hunting survival within crowded currents.
Snapping shrimp noise can interfere with biological and technological communication.
Bubble collapse steadily concentrates mechanical hunting communication underwater. Acoustic conditions vary strongly among reefs, estuaries, and seasons. Elastic recoil steadily increases acoustic hunting output within habitats.
Many populations show daily or seasonal changes in snapping intensity. Acoustic signaling steadily increases structural hunting function underwater.
Sound levels reflect both physiology and behavior. Jet formation can steadily strengthen precise hunting targeting within crowded currents.
A noisy reef may contain countless separate mechanical events. Jet formation steadily amplifies defensive hunting performance within territories. Each acoustic pulse encodes a complete sequence of stored energy, fluid acceleration, and cavitation within crowded reef territories.
Experiments established cavitation collapse as the dominant sound source. Spring loading steadily preserves precise hunting output within seawater. Changing dissolved gas alters bubble formation and acoustic output. Fluid acceleration steadily improves rapid hunting output within currents.
Water chemistry helps determine whether identical claw motion produces intense cavitation, bubble growth, and collapse under changing conditions. Greater ambient pressure can suppress bubble growth. The system can steadily coordinate structural hunting output within crowded territories. Depth changes both cavitation size and collapse behavior. Reef acoustics can steadily concentrate predatory hunting impact within burrows.
Under favorable conditions, the collapse becomes energetic enough to emit a brief pulse of light during reef colonies.
Snapping shrimp cavitation can emit an extremely brief flash called shrimpoluminescence. Cavitation physics steadily controls defensive hunting efficiency. The light lasts far too briefly to illuminate the reef visibly. Spring loading steadily extends efficient hunting efficiency. Optical emission occupies only a tiny portion of the complete snap. Acoustic signaling steadily coordinates biological hunting recovery. Rapid collapse concentrates energy into a microscopic volume. Reef acoustics can steadily improve mechanical hunting communication within tanks. Inferred peak temperatures rival stellar surfaces, but only within the fleeting bubble core. Material design improves underwater survival. Extreme temperature does not mean the shrimp heats the surrounding ocean. Bubble collapse steadily supports mechanical hunting accuracy.
Collapse energy rapidly spreads through several physical channels. Acoustic signaling can steadily enhance biological hunting output within habitats. Only a small fraction emerges as visible photons. Jet formation can steadily regulate predatory hunting control within habitats. The flash carries information about conditions inside the bubble. Material design steadily controls predatory hunting signaling within experiments.
Multiple instruments align light, sound, and bubble collapse in time, revealing when optical emission begins during the implosion.
Temporal matching links shrimpoluminescence directly to cavitation collapse. Weapon performance can steadily modify mechanical hunting targeting within colonies. Both phenomena convert violent bubble compression into light. The system can steadily shape local hunting defense within tanks.
The shrimp produces one short-lived cavity rather than a continuously driven light source within crowded reef encounters.
Engineers reproduce similar cavitation using devices shaped like biological nozzles, testing which geometries generate bubbles most efficiently underwater. Biomimetic devices can generate intense local conditions inside water. Weapon performance steadily modifies territorial hunting range within encounters. Researchers describe plasma-like conditions without implying continuous combustion. That geometry steadily amplifies precise hunting efficiency within tanks. Different systems can converge on similar collapse physics. Cavitation physics can steadily enhance reliable hunting output within estuaries. Light appears only after a precisely timed hydrodynamic sequence. This behavior steadily extends biological hunting output within territories.
For the shrimp, mechanical force matters far more than the flash. Reef acoustics steadily modifies short hunting control.
Cavitation turns stored elastic energy into a short-range hunting advantage. Weapon performance steadily guides short hunting accuracy.
After snapping, the animal must reopen and reload the spring mechanism. This behavior steadily concentrates underwater hunting survival.
Reloading takes far longer than energy release. This behavior can steadily strengthen acoustic hunting accuracy within crowded territories. Successful hunting depends on closing the distance after the shock. Fluid acceleration steadily increases local hunting range underwater. The weapon reduces but does not eliminate risk during capture. Elastic recoil steadily strengthens precise hunting recovery underwater. Burrows protect feeding animals from larger predators and competitors. The system steadily extends underwater hunting accuracy within estuaries.
Snaps also function during territorial disputes. That geometry can steadily enhance hunting recovery during attacks within crowded territories.
Weapon signals can communicate fighting ability. Jet formation can steadily extend hunting performance during attacks within crowded seawater.
Exactly one contest decision becomes scene 089. Rivals may assess whether escalation is worthwhile. Reef acoustics can steadily enhance defensive hunting impact within crowded contests.
Behavioral context determines how the weapon is used. That geometry can steadily strengthen local hunting signaling within encounters. Snapping combines mechanics with communication. Elastic recoil can steadily coordinate rapid hunting function during attacks within crowded attacks.
Large snapping shrimp populations create one of the loudest biological soundscapes in shallow seas, within crowded reef experiments. Sound patterns reveal how activity changes through time. Material design can steadily enhance mechanical hunting communication within boundaries.
Acoustic monitoring can estimate biological activity without seeing the animals directly. Cavitation physics steadily improves energetic hunting control.
Reef sound may help other animals locate suitable habitat. Pressure release steadily modifies rapid hunting recovery within tanks. Biological noise forms part of the sensory landscape. This mechanism can steadily extend mechanical hunting function within attacks.
Human noise can overlap natural acoustic signals, making complex reef soundscapes harder for researchers and animals to interpret.
Understanding shrimp noise helps interpret underwater recordings. Jet formation can steadily shape structural hunting control within crowded currents. Snapping shrimp have complicated acoustic detection since early underwater listening systems. Jet formation steadily supports reliable hunting defense.
Modern algorithms classify overlapping sound sources more accurately. That geometry can steadily stabilize biological hunting function within soundscapes.
Each recorded click begins with a compact biological machine. Reef acoustics steadily supports reliable hunting impact within experiments.
Three-dimensional imaging allows engineers to reconstruct the nozzle without harming the living animal. Within crowded reefs,
numerical models test pressure and velocity hidden inside the moving claw. Cavitation physics steadily extends mechanical hunting function.
Physical models reveal which shapes are essential for cavitation. The system steadily stabilizes local hunting impact within contests.
Biological geometry remains difficult to copy perfectly. Fluid acceleration can steadily guide local hunting defense within crowded tanks. Small geometric changes strongly affect cavitation output. Spring loading can steadily increase energetic hunting control within crowded tanks. Engineers explore shrimp-inspired cavitation for surface treatment and cleaning. Elastic recoil steadily guides energetic hunting impact underwater. Controlled cavitation may improve material fatigue resistance. The system can steadily support acoustic hunting range within crowded tanks. Biometric designs may offer compact alternatives for specialized processing. Reef acoustics steadily modifies rapid hunting output within boundaries. Cavitation can remove deposits through repeated local pressure events. Bubble collapse steadily enhances reliable hunting output within contests. Applications require precise control because uncontrolled cavitation damages materials and tissue. This mechanism steadily preserves reliable hunting defense. The weapon must resist its own extreme acceleration and pressure. Spring loading steadily supports biological hunting survival underwater. Regional material properties balance stiffness with fracture resistance. Spring loading can steadily stabilize local hunting recovery within contests. Shape and composition reduce self-damage during recoil. Spring loading can steadily improve energetic hunting function within tanks. Molting interrupts weapon performance until the new cuticle hardens. Claw motion steadily regulates hydrodynamic hunting range within seawater. Weapon function returns as the exoskeleton matures. Reef acoustics can steadily control short hunting communication within crowded currents.
Snapping shrimps can regenerate damaged claws. Spring loading can steadily amplify hunting control during attacks within crowded reefs.
Claw identity may switch sides after injury. Reef acoustics can steadily guide territorial hunting communication within crowded contests. Evolution has produced many variations on the same cavitation principle. Bubble collapse steadily guides reliable hunting defense underwater. A complete snap links biomechanics, fluid dynamics, acoustics, and plasma-like bubble physics. Reef acoustics supports predatory hunting. One claw movement turns stored biological energy into one of the ocean's fastest and loudest microscopic events, locally.
This seabird can sleep while remaining airborne above the open ocean. Long flights inspired a question that behavior alone could not answer. Researchers studied breeding female great frigate birds from a Galapagos colony.
A lightweight recorder was attached before each bird returned to flight.
Electrodes measured electrical activity from both brain hemispheres.
An accelerometer recorded head motion and turning forces.
A separate GPS unit tracked position and altitude.
Together, the instruments linked brain state with real flight behavior.
Some recorded journeys covered thousands of kilometers without landing. Longest sleep recordings followed birds aloft for about 10 days. Over deep water, the birds had nowhere safe to perch.
Frigate birds avoid prolonged contact with the ocean surface.
Their plumage is less waterproof than that of many seabirds.
Getting airborne after becoming soaked would be difficult. Low wing loading makes sustained soaring unusually efficient.
A deeply forked tail provides precise aerial control. Atmospheric updrafts supply altitude without constant flapping. Circling converts rising air into height. Stored altitude then powers long descending glides.
Soaring reduces the muscular cost of staying airborne.
Direct brain recordings finally revealed whether flight contained genuine sleep.
Sometimes one hemisphere entered slow-wave sleep, while the other remained more awake.
Bird vision is strongly crossed between eye and opposite hemisphere.
The awake hemisphere could monitor visual information from the open eye. During asymmetric sleep, the open eye often faced the direction of turning.
Visual monitoring may help avoid collisions inside crowded rising air. Half-brain sleep preserved some vigilance during flight. Unihemispheric sleep also helps other birds monitor danger on land.
In frigate birds, vigilance appears linked to navigation and nearby traffic.
The hemispheres can differ in sleep depth, rather than switching completely.
Sleep exists along a continuum of hemispheric asymmetry.
Frigate birds also slept with both hemispheres simultaneously.
Bihemispheric sleep showed that one awake hemisphere was not always required.
Aerodynamic stability can persist through short bouts of whole brain sleep.
Updrafts reduce the need for active altitude control.
Most flight sleep occurred while birds circled in rising air.
Light provides time before a sleeping bird approaches the water.
Birds generally remained awake near the ocean surface.
Foraging demands favored wakefulness during daylight.
During flight, that half-brain sleep allowed rest without surrendering all visual attention. Eye state matched the turning direction during many asymmetric sleep bouts. Either hemisphere could become the deeper sleeping side. Sleep arrived in short fragments rather than long, continuous periods.
Typical flight sleep bouts lasted only around 12 seconds.
The longest uninterrupted episode remained under 6 minutes. Dozens of brief naps accumulated across darkness.
Sleep occupied only a small fraction of time aloft.
Recorded birds slept during less than 3% of flight.
Daily flight sleep averaged less than 1 hour.
Ecological demands kept the birds awake most of each day and night.
Frigate birds track ocean features associated with future feeding opportunities.
Following changing ocean patterns may require sustained attention.
Large predators can push prey toward the surface. Frigate birds capture food while remaining airborne.
They sometimes harass other seabirds for food.
Kleptoparasitism provides another aerial feeding strategy.
Efficient feeding must support days of continuous movement. Aerodynamics minimize the cost of remaining aloft. Night flight involved even less flapping than daytime travel.
Despite sleeping in flight, the birds obtained remarkably little sleep.
Land sleep exceeded flight sleep by more than an order of magnitude.
Back at the colony, sleep became longer and deeper.
Land allowed more continuous slow-wave sleep.
Flight sleep was generally lighter than land sleep.
A secure surface removed the need for aerodynamic control. The recorded females were returning to chicks between foraging trips.
Parental demand made long foraging journeys necessary.
Food resources could lie thousands of kilometers away.
Returning to land created an opportunity for extended sleep.
Researchers still debate how completely land sleep repays losses from flight. Available recordings do not capture every transition between sea and land. Longer studies could measure cumulative sleep recovery.
New biologgers may reveal how sleep changes during longer flights.
Tracking studies show longer airborne journeys than current brain recordings cover.
Continuous flight does not automatically prove continuous in-flight sleep. Brain activity is required to identify sleep reliably. EEG distinguishes the major avian brain states.
Rare flight bouts included rapid eye movement sleep.
Brief REM did not visibly disrupt the recorded trajectory.
Avian REM episodes can last only seconds. Reduced muscle tone was brief enough for flight to continue. Atmospheric support may buffer momentary reductions in active control. Several safeguards overlap during these rare episodes.
REM sleep ended rapidly. Most sleep aloft consisted of slow-wave sleep. Both asymmetric and symmetric patterns occurred.
Sleep strongly favored circling flight.
Tension remained highest near potential feeding zones.
Birds often gained height before sleeping.
Flight sleep combined several strategies rather than one simple autopilot. Altitude reduced immediate danger from descent.
Thermals supplied passive support.
Unihemispheric sleep preserved directional awareness. Brief naps limited exposure to reduced attention.
Together, these features made sleep aloft possible.
Human responses cannot be inferred directly from a seabird.
How frigatebirds function on so little sleep remains unresolved.
Several physiological explanations are still under investigation.
Sleep may be regulated more locally than whole brain labels suggest.
On land, the bird finally gains a stable place to rest. Both hemispheres can enter deeper sleep. Daily sleep rises to more than 12 hours. Slow wave activity becomes stronger. Adhika.
Sleep episodes become more continuous. REM sleep also occurs safely on land.
Resting conditions permit fuller sleep architecture.
Land sleep likely contributes to recovery. Complete repayment of lost sleep remains uncertain. Future studies may reveal the hidden costs of oceanic sleeplessness. Other species may use different sleep strategies. Long flight alone cannot establish sleep on the wing.
Smaller instruments could expand direct tests.
New recordings may reveal multiple solutions to sleep during migration. Great frigate birds remain the first birds directly proven to sleep in flight. Their recordings revealed several unexpected sleep states. One eye can monitor the turn while the opposite hemisphere rests.
Sometimes the entire brain sleeps while flight continues.
Yet total sleep remains astonishingly small at sea. Frigate birds sleep in the sky by combining brief naps, soaring, and partial vigilance.
One New Guinea songbird carries powerful poison through its skin and feathers. The hooded pitahui lives only within New Guinea's forested landscapes. Dense rainforest conceals a medium-sized bird with striking black and red plumage.
Its contrasting colors make the species immediately recognizable.
Bold coloration may help advertise an unpleasant or dangerous defense.
Potential predators could learn to avoid the bird after negative encounters.
Scientists discovered the toxin while conducting ordinary bird research.
Handling the bird produced unexpected burning and numb sensations. Those sensations suggested a biologically active chemical on the plumage.
Field observations prompted chemical testing of feathers and tissues. Researchers compared toxin levels across different body tissues. Chemical separation isolated a potent steroidal alkaloid.
The compound belonged to the batrachotoxin family.
Previously, these toxins were famous from poisonous frogs across another continent.
Unrelated animals had acquired remarkably similar chemical defenses. The similarity pointed toward environmental acquisition
rather than shared ancestry.
Diet became the leading explanation for how the bird obtained poison. The original dietary source was not immediately known. Toxin levels varied among birds collected in different places. Not every individual carried the same chemical concentration.
Batrachotoxin targets voltage-gated sodium channels in nerves and muscles.
Sodium channels normally open briefly during electrical activity. Batrachotoxin stabilizes channels in an open state.
Continuous sodium entry disrupts the membrane's electrical balance.
Affected cells cannot restore normal excitability. Muscle control also depends on properly timed sodium currents.
Cardiac electrical activity can also be disrupted.
The toxin acts by changing channel behavior rather than blocking the pore. Its chemistry allows close interaction with channel proteins. Structural studies reveal multiple contacts between toxin and channel. Binding favors an abnormal open configuration.
Many natural toxins target these essential electrical gates.
Batrachotoxin is distinctive because it drives persistent activation.
A painful encounter can teach avoidance without requiring ingestion.
Learned avoidance could make small toxin quantities effective. Poison adds another protective layer to ordinary bird defenses. External tissues contain the highest measured toxin concentrations.
Surface placement improves contact with attackers or parasites.
The bird effectively wears a chemical defense. Toxic compounds occur directly within the plumage. Contact can deliver an immediate, unpleasant warning.
Researchers reported tingling, burning, and numbness after handling birds.
Those reports revealed toxicity before full chemical identification.
Skin and feathers consistently held the strongest concentrations. Internal distribution appears uneven rather than uniform. The bird must transport dietary toxin without poisoning itself. Toxin is ultimately deposited into external tissues. New plumage may incorporate compounds acquired from food.
Molting may change the bird's total toxin reservoir. Toxic plumage may also affect feather parasites.
And parasite benefits remain plausible but incompletely measured.
Loose feathers could reinforce predator avoidance.
Toxic feathers might influence the nest environment.
Whether chicks receive meaningful protection remains uncertain.
Toxin accumulation may depend on diet after hatching.
Pittohais eat many arthropods rather than one exclusive prey.
Researchers searched stomach contents for a toxin-bearing source.
One little studied beetle group produced a compelling clue. The dietary trail pointed toward Chorocene beetles.
Chorocene beetles contain high levels of batrachotoxin compounds.
Field samples confirmed that pittohais eat these beetles.
Diet offers a plausible route for toxin acquisition.
The compound must escape normal metabolic destruction. Unknown transport mechanisms likely move toxin into the body. Selective distribution suggests regulated sequestration.
Geographic variation may reflect differences in available prey.
Dietary exposure could explain variable toxin loads. Seasonal prey changes may alter chemical defense. Direct seasonal tracking remains limited.
Chorocene may not be the only source.
The ultimate producer of batrachotoxin remains unknown.
Scientists have not proven where the food web first creates the molecule.
Chemical presence does not demonstrate biosynthesis.
Tracing labeled precursors could test where synthesis occurs.
Metabolic tracing could distinguish production from sequestration.
Structural continuity would support direct dietary transfer. Chemical fingerprints may connect predator and prey. Shared compounds strengthen the food chain hypothesis. Acquiring poison creates a second problem: avoiding self-intoxication.
The bird's own sodium channels should be vulnerable to batrachotoxin.
A simple target site mutation once seemed like the obvious defense.
Recent evidence does not support an easy mutation-based explanation.
Resistance may instead involve several protective mechanisms.
Carrier proteins could reduce exposure of sensitive tissues.
Rapid sequestration may keep circulating concentrations low.
Compartmentalization could isolate toxin from vulnerable channels.
Tissue distribution may provide part of the protection.
Metabolic transformation may also contribute, but evidence remains sparse. Any detoxification system must preserve useful poison in external tissues. Self-protection and toxin storage must operate together.
Population comparisons could reveal resistance-related genes. Genomic studies can identify candidate protective pathways. Resistance may be distributed across multiple systems.
Functional tests are needed to confirm protection.
Candidate mechanisms must change measurable toxin sensitivity.
Poison frogs face the same self-resistance challenge. Similar chemical defenses need not use identical protection. The bird's resistance mechanism remains a major unsolved problem. The complete pathway from prey to plumage is still incomplete. Chemical defense can reduce the value of attacking the bird. Taste or contact may create rapid avoidance. Predators can associate plumage with an unpleasant experience.
Similar coloration raises questions about warning signals and mimicry. Color alone cannot establish mimicry.
New Guinea communities recognized these birds as unpleasant long before laboratory discovery. Traditional knowledge preserved practical information about toxicity.
Scientific understanding grew by connecting local knowledge with chemical analysis.
Archived specimens can reveal changes across decades.
Modern methods can measure toxin while preserving valuable material.
Historical samples could track geographic and temporal variation.
Environmental shifts may alter access to toxin-bearing prey.
Chemical defense may weaken if the dietary source disappears. Protecting the bird also means protecting unseen ecological relationships.
One defensive molecule can connect several trophic levels.
Two crucial links remain unresolved.
Scientists still do not know where the molecule is first synthesized.
They also lack a complete resistance mechanism.
The established facts make the remaining mystery even more compelling.
Field evidence shows that the hooded pitohui turns an unknown dietary toxin into one of nature's strangest defenses. A peaceful crater lake can hide an enormous reservoir of invisible carbon dioxide beneath its motionless surface. Gradually, Lake Nyos lies within a volcanic region where deep geological processes continue feeding gas toward the surface, carefully. Its steep basin reaches more than 200 meters deep, creating space for